Everyone who's currently watching for coming and watching, and secondly, I'd like to thank Jeremy and Sammy, who have both been fantastic. I think we'd all agree in organising these events. It's been a fantastic series so far. So I'm going to talk to you today about a new supramolecular technology I've been in charge of innovating since I joined the University of Kent back in 2015. And this technology is based around developing novel antimicrobial weapons to as antimicrobial resistance becomes more prevalent in our society, to combat that. And what I'd like to propose to you today is the ability to take these practical data sets and try and look at how to incorporate machine learning and artificial intelligence to drive these technologies forwards. So bridging that gap between machine learning and artificial intelligence and those results that we get as practical scientists within the biology and chemistry laboratories. So first of all, I should probably tell you a little bit about the University of Kent, because not many people have heard of it. We're closer to France than we are London. We're in that part of the UK, and we're a fantastic historical city centre with an awful lot of British pubs and a very hip and down with the kids unicorn type pub here. So like if anyone's ever in the vicinity of Canterbury and wants to drop in after obviously the pandemic has all died down, we'll be more than happy to come and say hello. So first of all, because of the breadth of the audience, I'm just going to give you a little definition of what supramolecular chemistry actually is. So in supramolecular chemistry, we're not so concerned with making covalent bonds. What we're concerned with is non-covalent interactions, things like hydrogen bonds and ion pairs and the such. And this area of chemistry is a very young area of chemistry. The term supramolecular chemistry and the description chemistry beyond the molecule was first coined in 1987 by Jean-Marie Lane, who won the Nobel Prize for his research in this area. And you can think of this type of chemistry as the chemistry of Velcro interaction. So we've got two molecules, like the two sides of the Velcro coming together, getting stuck a little bit and then being pulled apart again, although those two sides of the Velcro stay intact during this process. And that's the kind of chemistry that I'm really interested in and my background. So the research that underpins this, this presentation is based around this group of publications here and this list of patents that we have here. Obviously this is an antimicrobial technology, so we have moved to patent it in this instance, but we've obviously published an awful lot of it as well. And it's all based around this very simple molecular construct, all of our technology. So we've got this hydrophobic group here, this phenyl ring system. Then we have a hydrogen bond donor, hydrogen bond acceptor system here. This is termed a urea or a thiourea group, but is illustrated for the purpose of this cartoon as a purple shape here. Then we have this alkyl spacer here, and then we have an anionic functionality here, typically a sulfonate or a carboxylate. And finally we have this countercation that's to the anionic group on this monomeric unit structure. And what's interesting about this from a supramolecular chemistry point of view, and the reason we ever started looking at these things in the first place, is because although this is a very, very simple molecule, it can self-associate in a number of different ways through the formation of hydrogen bonds. We can get this, you could think this dimer formation, two of these coming together through the formation of 4 hydrogen bonds, or we could get this end-on tape formation between the anion and the urea or the thiourea group, or we could have this urea-urea or thiourea-thiourea stack motif with the anionic units and alternating sides, or we could have this syn stacking where we've got these anionic units on the same side. But does this happen in real life? And we're quite pleased to say we have some evidence that it does. So these are 3 single crystal X-ray structures of the same anionic unit. And we see all of these binding motifs represented here. We've got the formation of this urea anion dimer here. We have the formation of this urea anion tape here. And we have the formation of this urea-urea syn stack here. All the same anion, exactly the same, but in this case we're controlling the different self-associated modes with the countercation. Here we've got a strongly coordinating countercation, sodium or potassium. Here we have more weakly coordinating countercations, in this case pyridinium, and finally we have tetrabutyl ammonium, the weakest of them all, which results in this dimer formation. And we've also shown that beyond the supramolecular chemistry, this class of compound acts as an antimicrobial agent, or a source of them. We have over 70 now. But why should we care about creating new antimicrobial agents? Well, because there's a growing need. These are images taken from a review on antimicrobial resistance, published in 2014 by Jim O'Neill. And it shows the current state of antimicrobial resistant infections and around the globe. So in 2014, 700,000 people around the world per year were dying from antimicrobial resistant infections. This is a lot less than those leaders say counts here, 8.2 million deaths. However, by 2050, this number is going to rise. It's predicted to become 10 million deaths per year. That means death by antimicrobial resistant infections is going to become more prevalent in our global society than cancer. And these are just the primary effects. Those secondary effects are going to prematurely end the lives of 300 million people per year around the globe by this year 2050, or at least that's what this predictive data tells us. This is also going to have a financial effect. If you don't care about the effect on human life, then there's also a financial effect. It's going to cost the world's GDP $100 trillion by 2050. That's 67 trillion pounds. Where are the effects going to be most felt? They're going to be most felt in low and middle income countries across Africa and Asia, with the highest death rate per capita. In Africa, that's 4.15 million deaths per year in 2050 from antimicrobial resistant bacterial infections. And because there isn't a great amount of money to be made out of the development of novel antibiotics for those big corporations, it really does come down to the likes of ourselves in university and small industry settings to pick up the baton here and start to push these technologies forwards, which is what my research is all about. So when we first discovered that this type of compound could act as an antimicrobial agent, the first thing we thought about was, okay, we've got one hit, how do we make it any better? So we've actually published to date 50 different variations of this structure. Okay, we've got this I'd say our beginning structure here, and we've basically changed everything we could think of. We've changed these R groups here with electron donating and electron withdrawing groups. We've changed this X here for an oxygen or a sulfur. We've changed the length of this spacer here. We've changed the anion here, and we've changed the cation. We've also played around with adding extra ring systems off the back of it, which also is very handy as it makes some of our molecules intrinsically fluorescent, so we can watch them down a microscope basically to see what they're doing. And then we've got rid of these ring systems altogether and just add alkyl chain units, got rid of this urea system here just to have this anion, and then just explore the antimicrobial effects of our cationic components as controls. But to add to that complexity, it's not just about the antimicrobial activity of those 50 compounds, but these compounds have the ability to self-associate, not only self-associate, as we showed on the previous slides, with just single molecules, but to form different types of materials. So this same single molecule here, if you stick it in organic polar solvent, will produce a dimer. You stick this same molecule in water and it produces a spherical aggregate species here, and you add salt to that water, heat it up, cool it down, and these spheres morph into strands and you end up with a supramolecular material. So not only how do we have to investigate the antimicrobial properties of these things, we have to understand what these molecules are doing both at the material level and the molecular level in order to understand their structure activity properties in order to drive antimicrobial development. So this means that every time I send one of my students into the laboratory with a, can you just make this new molecule? First of all, they have to make that new molecule. They then have to characterize it using proton, carbon, IR, melting point, mass spectrometry, and in silico modeling. We do a lot of simple PM6 semi-empirical modelling to get electrostatic potential maps to look at surface charge distribution. They then have to go away and produce crystals of these materials and perform X-ray crystal diffraction studies on these things so that we can get those nice pictures to show the self-associated modes of these different molecules in the solid state. They then move on to quantitative NMR experiments so we can figure out whether or not when we dissolve these molecules in different solutions, how much of that molecule ends up in these bigger structures and how much is just left freely floating around. If there are bigger structures, then we're looking at characterizing these materials by dynamic light scattering, zeta potential, critical micelle concentration, scanning electron microscopy, transmission electron microscopy, and fluorescence microscopy should the material be fluorescent. If we don't see any evidence of these bigger structures, we've still got just as much work to do. We have to look at evidence of slow exchange on the NMR timescale, which is a bit of a pickle and can lead us to do variable temperature NMR. We then have to look at producing, figuring out how strongly those molecular units may come together to form these smaller structures. So we're looking at conducting proton NMR self-association studies, and coupled with that, we have to do diffusion ordered NMR spectroscopy. But even conducting these techniques, although there's a lot of them, there's a lot of learning to be done there by an individual. It's also a very time-consuming process. So this is the data that we have to collect if we want to calculate just one molecular self-association constant. First of all, we have to take a solution of our molecule, we have to stick it in some NMR solvent, stick it in an NMR machine, and then dilute that sample. So each one of these spots here is a different NMR spectra. We then have to plot it and take this data and fit it to a number of different binding isotherms. Thankfully, there's some online platforms to enable you to do that these days, but to figure out what kind of self-associated processes are being undergone in solution, we fit them to different binding isotherms, and sometimes it's really easy to pick out which binding isotherms your data is fitting to. If we look here at molecule 28, we've got a 1.5% error and a 0.8% error for this EK model here. However, for co-EK models, we're looking at 43, 21, and 47% error. So we're likely that our data is fitting very nicely to this model here, and it's a complete disaster when we look at it fitting at this model here. However, with other data, fitting is not so clear cut. For example, molecule 31, we've got 0.6% error here and 0.3% error on our fittings here. However, we have 0.7 and 0.3 and 2% error on that alternative binding model. So this data fits quite nicely to both binding models. So it's at this point you have to turn to things, other techniques such as DOSY, to try and figure out the size of your self-associated species in solution to be able to provide evidence for quoting one fitting over another fitting. Moving into from just the molecular self-association into the material characterization. If you've got fluorescence compounds, you can use fluorescence microscopy and actually gain images of your self-associated species, and you start thinking, great, that's all I need to do to characterize what on earth I've got in solution. Well, this is not quite the case, because your eyes are drawn to what your eyes want to see. So when we're looking at characterizing what we have and measuring the size of those self-associated species in this case, so you just put a little measurement, you measure this on the screen to figure out how big those self-associated spherical aggregates are in this case, you tend to just measure what you see, and that's not always the best scientific approach. So you end up then using complementary techniques. So this data is for this purple molecule here and this molecule shown in blue here. Those empty bars here are the data that we collected from our fluorescence microscopy techniques, and here these filled bars are the data we collected from dynamic light scattering techniques. And this is not the error on our measurement. This is, in fact, the spread of the sizes of those species identified by either technique. And what we see here is, in this case, we've got good agreement here. The average size of those molecules, of those self-associated aggregates, sorry, that we identify by fluorescence microscopy and by dynamic light scattering are similar in this instance. However, moving to some other species, and it's not quite so comparable. definitely picked out what we wanted to see here, for example. But yeah, so we need more than one technique, and they need to be complementary to one another. If you do get these self-associated species, when you see what they are, you also need to know something about their formation and cue critical micelle concentration determination, which is also a parameter that's used a lot in drug development, to guide drug development. And these experiments to elucidate the critical micelle concentration values for a specific compound are also pretty personnel heavy. So we do this typically by measuring surface tension of a solution with our compounds in at different concentrations, plotting that data, and where the surface tension then ceases to decrease anymore with increasing concentration of our compound in solution, this crossover point as we've shown here, this is the critical micelle concentration, or what we define as a critical micelle concentration. And this looks very pretty, but in actual fact, what's happening is a student is sat there sucking up solution into this syringe, dropping out one drop at a time and measuring the angles of this drop and using that to calculate surface tension. And they have to do that 3 times in order to get a single point on that graph that we showed on the previous slide. So it's a pretty, again, time exhaustive process there. And it's at this point that I'd really like to thank these individuals that have spent so long creating the data that I'm overviewing for you today within the scope of this presentation. And when I was thinking about this, and the reason I've put the slide of these individuals in right now is because I started thinking exactly how much time have we taken to characterize these 50 molecules? The answer is 31 years. So 31 years of these students' lives and my own life, I've contributed my own work time to this effort, have been taken to create the data set that you're seeing here today. This is an incredibly expensive process. When we're looking at characterizing these self-associated processes and producing what we consider to be quite an expansive data set, but quite a few of you might be sat there going, she hasn't even got a drop in the ocean yet. So we've characterized the self-associated properties behind these molecules because we now know from the experiments that we've conducted that they're incredibly important towards designing ever more active antimicrobial agents. But the other side of the story is how these things work, these molecules work when they come into contact with the bacteria themselves. And what we now have quite a little bit of evidence towards is the fact that these molecules are able to selectively interact with different types of phospholipids, and those phospholipids are those molecules at the surface of your cells and indeed of bacterial cells, but they differentiate depending on what type of bacteria or whether you're a eukaryote or a prokaryote or whatever. So these are bacterial phospholipids shown here, or the structures of bacterial phospholipids shown here, and this is a structure of what is commonly classed as a human phospholipid. And we can see here we've got some not so preferential interactions. And we've also been lucky enough to show how the self-associated properties of these molecules, their self-associated forms, may interact with the surface of bacteria to help with this antimicrobial mode of action. So here is an image I showed you previously of one of our self-associated spherical aggregate structures, and this is what happens when you take a solution of those spherical aggregate structures and inject them into a culture containing, in this case, methicillin-resistant Staphylococcus aureus, our good old friend MRSA, the plague of our National Health Service. So we have these spherical aggregates, and what we can see here is that they have bumbled along and they're starting to adhere to the outside of the bacteria. Leave it for 30 minutes, and in yellow this represents E. coli bacteria, and the red flowery things represent MRSA here. And what we see using fluorescence only microscopy in this case is that there's no longer any form of spherical aggregate structure. Indeed, these spheres have now splattered themselves all over the surface of our microbes. So here we have E. coli and here we have MRSA. Okay, leave these molecules in contact with our cells for a little bit longer, up to 4 hours. This is a mixture here of methicillin-resistant Staphylococcus aureus and E. coli. And you can see that although these molecules are still adhering to the outside of that cell membrane, they're also starting to internalize and end up in the center of the cell. So basically what we've got evidence for so far is that these things self-associate into these spherical aggregates. In this case, they collide with the surface of a bacteria, splat themselves all over the surface, creating this coating. They then interact with those phospholipids at those surface, we hypothesize, and then burrow their way through the membrane. We know they actually get through the membrane to get to the other side, because those, in some cases, cells carry on growing even though we know that these things are internalized, where they cause all kinds of merry hell. So this is a very complicated process for such a simple looking molecule. This is not a simple just drug molecule binding in a specific receptor site. In this instance, it's far more complicated than that. But the good news is, for the 50 compounds that we've published to date, we do have a good range of antimicrobial activity, and this can help us maybe derive those structure-activity relationships. Here we see for MRSA, this dotted line here, this black dotted line here, is the normal growth of methicillin-resistant Staphylococcus aureus. And this coloured lines here is what happens when we add a comparative concentration of different candidates from our library to that microbial solution. And you can see here we've got compound 15 that's pretty terrible, to be honest. It's not really impacting on growth whatsoever. And then we have this yellow line here, this compound 35, and it's doing an okay job. It's not brilliant, but it's doing something. And then we have these green compounds here, compounds 1 and compounds 4, that aren't even allowing those microbes to begin to replicate within that microbial broth. Moving on to E. coli, so MRSA is a gram-positive. They're relatively easy to kill comparatively to those pesky gram-negative bacteria. And those gram-negative bacteria are a bit of a pain because they have a double layer on the outside of their cells that these compounds need to get through and disrupt. Okay, so there's quite a lot more material for them to tackle there. But again, we see a range of different activities. Again, this black dotted line is the normal growth of E. coli. And again, these coloured lines are what happens to the growth of E. coli when we add in our compound. And we've got a good range again here. We've recently been able to enhance the activity of these compounds with some new candidates. So we do have some just flat lining. These are called growth curves. But these are not yet published. My students will not let me have the data yet. I just have a photo on WhatsApp. But looking at the data here, what we see is that compound 1 and compound 4 are doing a pretty decent job at preventing the growth of E. coli here, whereas with this compound 35, not too great, but doing something, and compound 15 again is a complete disaster. So we've got a good range of structures with different antimicrobial activities there. So to present all this data in one slide, which is heavy going for the best of us. What this slide represents are the antimicrobial efficacy results for 50 of these compounds. So these are our minimal inhibitory concentrations needed to inhibit the growth of 50% of that bacterial population. And we've plotted it as 1 over the MIC50. Basically, the higher these bars are, the better the antimicrobial activity of these different compounds is. And what we've tried to do is plot the results for each one of these compounds with just a stepwise change to that molecular structure. So although there's no numbers here, we've just made a single stepwise change here. And we can see that that has resulted in some pretty nice structure activity relationships just going on here with MRSA. And we've also got some nice candidates that have a similar activity against both gram-positives and gram-negatives, so our MRSA and E. coli here. And we've also got some really lead compounds that are literally, well, quite a lot more effective than some of our other compounds. So again, identifying themselves as lead compounds. So we've now seen that, I've now told you that, these molecules are interesting to study just from a supramolecular chemistry point of view. I've told you that the self-associative properties of these molecules are vital for their antimicrobial activity, or at least that's what our results to date would show. We've shown that the selective interaction of these molecules with the surface of those membranes is also very, very important. But to add one more problem to the mix, we also have multiple materials. Remember, this same molecule can exist as a dimer or a spherical aggregate, or morph into a hydrogel. Okay, so we now have to ask ourselves, the supramolecular chemist, making a new material is really super cool, but is it worth including in our antimicrobial activity program to take this technology forwards? So just to prove to you that this is a supramolecular gel, and we have undergone this transition from sphere to strand. Here we have some preliminary investigations. These are the results of the investigations we use to see whether or not we have any plausible material form whatsoever. So we take our molecules and we dissolve them in our solution. Here we've got a water control. Here we have a sodium chloride solution. Here we have a sodium phosphate solution. And here we have a mixture of sodium chloride and the antibiotic ampicillin. And what you can see is when we invert these tubes, which do contain a solution and our compound, we can't see any solution or compound in this H2O control. That's because we have our spherical aggregate. But when we have the formation of a material, we see the material clinging to the bottom of this of these vials as they are inverted, meaning that we should investigate them for possibly forming a supramolecular gel. The next thing is to characterize, to see if we actually do have gel fibers, and then to go into the physical properties of those gel fibers using, or that material, sorry, using rheometry, which is something that we have done, but I'm not going to overview for you today. But we can have a look at the formation of these gel fibers. So this is an SEM image where we've taken away all the liquid, so all that's left is the solid construct, and what we see is these fibrous networks. These are the ends of those fibers, these dark blobs here. So you're kind of staring down the fiber at that point. But we're also lucky, again, this is an intrinsically fluorescent molecule, so we can actually visualize those strands that are formed, those gel fibers that are formed in the solution state using fluorescence microscopy. And this is the beautiful structure that we managed to obtain. And I can literally sit here staring at that for hours. We were all very excited when we saw that. Where we don't have the formation of a material, though, this total gel formation, and we get something in the middle that looks like a very viscous, sloppy liquid thing, a bit like your kid's jelly that hasn't quite set right. And we look at that under SEM and the fluorescence microscopy. What we see are these kind of images here, and I don't know if you can just see, but we've got these spherical thingies here in our SEM images, and in our fluorescence microscopy images, we have some kind of strand formation, some kind of fiber formation, but it's when you watch it live, you can see all these little spheres shooting around these fibers. So we've got this intermittent stage where we have both the gel fibers and the spherical aggregates, but we don't have complete conversion, so the material is not stable. We don't have this supramolecular gel formed. These gels have some pretty nice properties as well when you look at perhaps utilizing these in contact with skin, say. We look at our sodium chloride gel. We've got the pH of this gel solution about 6.23. It's not going to irritate your skin too much. The melting temperature of this gel is 51.23 degrees Celsius, so storage not going to be too much of a problem. These things are held together by non-covalent interactions, which are susceptible and fall apart with heat. Remember, so keeping things stable up to 50 degrees C is quite a good thing. And also the minimum gelation concentration is very low. So this is the amount of material in milligrams needed to gel 1 milliliter of the solvent that it's stuck in. And 1.5 milligrams per milliliter is pretty good going there. But the next question is, in this self-associative form, do these molecules maintain their antimicrobial properties. We've seen them do it as a sphere. Can they do it as a strand? And the answer is yes, they do maintain their antimicrobial properties. So here we have a dollop of our hydrogel, our lead hydrogel with sodium chloride, on this agar plate here that's been inoculated with bacteria. And where you see these halo effects here, it's where the gel has stopped that bacteria from growing. With MRSAs, pretty good for a gel actually, being that it's a solid lump. With E. coli, not perhaps quite so convincing. So what we did was we incorporated the antibiotic ampicillin within this hydrogel, and we got a much bigger zone of inhibition going on here where there's just no bacteria growing whatsoever. So we all got quite excited about that. So we wanted to know whether or not that was the strand plus ampicillin doing the work, or if it was just the ampicillin by itself with the gel fiber there as kind of a delivery material, if you like. So we conducted these well studies where we core out some of this agar, shove our material in it, and then see if we get a zone of inhibition. And the gel by itself, nothing really to report, but with the ampicillin present in the gel, we get this big zone of inhibition, which is very similar to a zone of inhibition where the ampicillin is just in solution. So we think the ampicillin is diffusing out of the material at that point. But we wanted to then finally get into our heads whether or not supramolecular chemistry principles were important when we're trying to design next generation, ever more effective molecular technologies towards being developed as next generation antibiotics. And the answer is, through this quick study, yes, we have managed to show that. So here we took a series of 3 molecules. Okay, we've got a very electron poor version of our molecule here. This is an anthraquinone, and here we have an electron rich version of that same molecule with an anthracene. And when you take these 2 molecules and you shove them in a pot and you allow the liquid, you dissolve them, allow the liquid to evaporate, and if you're lucky enough to get single crystals, as we were, and you analyze the structure of the self-associated species within that single crystal, all we were able to find was these unsymmetrical dimer formations. So these things are selectively picking each other out, which is fantastic. But what does that mean for antimicrobial development in this case? Well, this is the normal growth of MRSA, this dotted black line here. This red line here, this is in the presence of just our anthraquinone molecule. This yellow line here is just in the presence of our anthracene molecule. And when we halve the concentration of our anthraquinone A, when we halve the concentration of our anthracene B, shove them together, so same number of molecules as the previous studies, but 50% of A and 50% of B, this is the result of those antimicrobial studies. We can see a lot less microbial growth. So playing around with the supramolecular chemistry does appear to produce more effective antimicrobials in this instance. So in summary, what do we have? We have a multifunctional tailorable system that we're able to tailor in the solid state through changing the cation. We've got anionic units that are able to dimerize in the gas phase and with an organic solvent system such as DMSO. Stick those same molecules in aqueous conditions and we have compounds that exhibit surfactant type properties with spherical aggregate formation in the range of 100 to 550 nanometers. Add some salt to that and we get a triggerable change from spherical nanostructure to strand, and we retain the antimicrobial activity against both MRSA and E. coli in either material form. And additionally, recent results that we've just published have shown that the hydrogels and the spherical aggregates also show promise as drug delivery vehicles and materials, which is quite exciting. So through all of that, each one of those 50 molecules that we have at least 16 parameters calculated for them. We've got molecular mass, well, that's a pretty easy one to obtain, but we've got things like verification of dimerization in the gas phase. We've got things like dimerization constants in the solution state. We've got things like the size of self-associated aggregates, stability of those self-associated aggregates, critical micelle concentration, surface tension measurements, the percentage of a compound that ends up forming these larger self-associated species. A whole raft of computationally derived parameters from electrostatic potential maps using PM6 semi-empirical modeling. And we have our concentrations needed to inhibit certain proportions of the growth of both MRSA gram-positive and E. coli model gram-negative bacteria. So with this huge amount of information, or I perceive as a huge amount of information that I have taken 31 years of people's chemistry lives and biology lives in order to obtain, we got quite excited about approaching some form of machine learning and artificial intelligence to produce ever more effective systems. And the supramolecular chemists that I'm friends with were really excited about this. Yeah, that's absolutely possible. I went and spoke to some machine learning people and they were, well, we could potentially look at it, but there's a lot of limited data here and it's only 50 and we need thousands. And I literally had a heart attack at this point. So how do we solve these issues? Okay, so the first thing we tried to do was maybe we could find some trends in our data that we already have and use that to kind of predict what kind of physical properties those molecules would have. Maybe that's a good place to start because we don't really have any other options right now. So what we did was we took our antimicrobial data and we started plotting that against just a single one of our parameters. And for E. coli, we came up with quite a nice trend against critical micelle concentration. This might not be a surprise to those that are familiar with developments in this area. We do have this big outlier, and this is fantastic because this molecule being an outlier makes me believe what I'm seeing because it is very structurally different. It is a control compound to those other compounds included within this study. So fantastic, bit of an easy hit for E. coli. The problem came with MRSA. So when we tried fitting against one parameter, it all just went a bit gobbledygook, and I was then faced with 14 parameters and not knowing really what to do with them. And it's at this point we started working with Dominic Chu over in the School of Computing here at the University of Kent, and he said it's really simple. All you do is an exhaustive search of models. We just take these equations that you think, as I identified these equations as being something that was a good starting point, and he said we just run them through on this automated R software. And I started going, I've only really learned how to use the internet, I can't use this, but he's very patient with me. So we started using exhaustive search methods to see if there were any trends within our data. And we started to pick out a few. I mean, they're not great, but there's a few things, a few trends that started to spring out when we started fitting these things to linear fits. We've got some outliers, but this is our lovely outlier that we get relieved every time we see it's an outlier. And a few others that we can explain chemically, actually, as to why they might not adopt the same mode of action as perhaps some of those other compounds that we're studying. And then we had the same outliers pick up again, and we got a little bit more excited. Maybe there are some links here. And again, but the take-home message of this to us came for the types of parameters that were being picked out by these exhaustive search methods to best fit this linear fit. And that was that we always had this critical micelle concentration value in there, and that we always had values to do with aggregate stability, and that aggregate stability comes from the supramolecular chemistry and the strength of those interactions. We'd already previously shown that. So we became quite excited about this. There may be some links there. Okay, so the other big problem is if we need a more extensive data set, we've got to work out a quicker way of getting at all these parameters, because right now it's 6 months per compound is just simply not viable for anybody's sanity. So we started developing novel assays to try and look at things in a more high throughput way. And the first was to kind of pick apart streamlining some of this self-associative structure determination. Can we at least get to that? So we started working with a plate reader company, and a plate reader was something I bought so we could all do our antimicrobial studies within the lab. And it's brilliant. You have these little plastic cassettes. They've got kind of holes or wells in. You can get up to 384 samples on a single plate that you just shove in a machine, program, press go, and it goes and analyzes 384 samples in a shot. This is much better than one. Okay, so we started looking at optical density measurements of this compound that adopts dimers in one solution, spherical aggregates in a second solution, and gels in a third solution. And we could see really clearly differences when we had just a dimer formed, as to when we had spheres formed, as to when we had these gel fibers formed. So we went from being able to screen one sample in a couple of months to screening 384 in an overnight thing whilst we were all in bed and asleep, and that is the best way of gathering data when you can leave it to gather itself. But in addition to that, we did manage to derive some physical chemical properties. Now, previously, when we were trying to figure out the melting temperature for our different gels that we'd formed, it involved a PhD student sat there with a heated plate, putting the material on the heated plate, setting that to a temperature, and then figuring out whether or not it had melted or not. This is not a sensible way forward. It's very time consuming and not the best scientific approach. There's a human error that can sneak in there. But using, observing the optical densities at different points in these little wells within this big plate via these plate reader methods, we began to see something quite interesting. So here is, we can split these wells down and we can analyze 170 different points in one well without having to be there, and we can do 384 wells in a shot, so this is fantastic. We started measuring the optical density at different points in the well, so that's and comparing these values. And basically what you need to take home from this is that we use a color coding scheme. Dark red is a high optical density, then we move through to pink, then to light blue, then to dark blue, and we have low optical density. And where there's big self-associated structures there, it increases the optical density measurement. So you move into those pinks, move into those reds. Where there is no really large self-associated structure present, we get these blues. Okay, so as we are melting our gel, disassembling those fibers, we move from red through to pink into blue. And through ratioing the highest optical density measurement and the lowest optical density measurement in a specific well, we're able to pick up those melting temperatures. I cannot tell you how painful this is before we started doing this. Okay, so here is a gel that we previously characterized to have a melting temperature of 51 degrees centigrade, and you can see that when we ratio the optical density measurements, the highest one in that well and the lowest one in that well, they stay pretty much the same as we increase temperature, but then the gel's not melting until 51 degrees, so this makes perfect sense. When we have a gel that melts, and this second gel melted at 43 degrees centigrade, as we heat up the sample, we can see that as we ratio the highest optical density measurement in that well with the lowest optical density measurement in the well, we start to see a decrease in this ratio here. And it just so happened to completely drop off between 40 and 45, and we know that the melting temperature for this system to be 43. So this method's looking pretty exciting as far as we're concerned for being able to increase the throughput of our characterization of our system so that we can access more data, but not in 6 months per compound. The last thing that we had to kind of add to this data set is the strength of interaction of a compound with different types of phospholipids, so there's molecules at the surface of the cells, and in order to add that as a parameter into our data sets. But trying to figure that out is absolutely a pain. It requires specialist equipment, it requires specialist training quite a lot of the time. It involves going to synchrotrons and things like that, which I don't know how to work a synchrotron. So we came up with another assay that uses equipment that most chemistry and biology equipment across the world have. A solution state NMR spectrometer. You can get away, we did these on a 600 megahertz machine, but I have a feeling you could get away with these on even a 400 megahertz machine, this type of experiment. So what are nanodiscs? Nanodiscs are basically Pringle-shaped 10 nanometer wide discs of phospholipid that are belted around with a synthetic molecule. And this synthetic molecule is a byproduct of the paint industry. They sell it in ton loads. It is as cheap as anything. So you buy this polymer, you stick it in with either your cells or your vesicles or whatever type of phospholipid containing parent structure you have, and it goes along and cookie cutters out all these little nanodiscs, and these little nanodiscs you can shove in your solution, okay, and then you can run studies where you look at how your molecules interact with these different types of phospholipid. And from that, through very simple, just literally figuring out the area underneath the curve and plotting that, you can start to figure out how strongly your different molecules coordinate to different types of phospholipids present in that sample. So here's an example. So here we have 3 different versions of our molecules here. Here we have some this is an experiment with some E. coli nanodiscs, and we can see here that we get an immediate response, a very low nanodisc to SSA ratio, so SSAs are our molecule for this compound here. It sticks like no tomorrow to these E. coli derived nanodiscs with those phospholipids in. However, these other 2 compounds here are not quite so good. So we've got a way of getting at those phospholipid molecule interactions, and these things can be programmed into an NMR machine. So you just make up your samples, stick them in a rack and press go again, and then go off and do other things whilst you're accumulating your data, which is cool. So the other way of looking at it is, are there any other data sets that we might be able to add to our own data set in order to get somewhere? Because as we bring multiple different data from different streams together, perhaps we can build something bigger that enables these type of technologies to move forwards while not taking all the work on ourselves. I am well informed that extracting information out of supplementary information papers is a pain in the neck, and that web scraping and such techniques can get you so far, but as I'm aware, and I'm in no means an expert in this area, it's also fraught with difficulty, and then trying to do something with the data after you collect it in multiple different formats is also difficult. So curating the data, that's the word, is also difficult. So that's coming along, but is there something we can do in the meantime? Well, perhaps there is. There are 2 different large data sets that I know about as a supramolecular chemist, and someone that's interested in antimicrobial technologies that I haven't seen brought into the scope of such studies such as this. And the first one is a platform called BindFit. And this is where I get the name wrong, sorry if I do. Paulie Thoranderson developed this platform, and it is basically a platform that allows you to take your dilution study data or your titration data from either UV-Vis or NMR or whatever else techniques you use, and to fit these data to binding isotherms. So where you've got molecular self-association and you're looking for association constants, or you've got 1-to-1 complex formation, 2-to-1 complex formation, host-guest 2 molecules coming together. You can run those studies, upload the data into this platform, fit this data to get out your association constant, but that data is then saved in that platform under a permanent URL. And pretty much every big supramolecular chemistry group that I know of now places their data within this platform. We're talking across the UK, US, and also in Australia. It's kind of the norm. So now, instead of finding vast amounts of how did I fit this data, and yes, I made my own, fitted it to my own isotherm, it's a URL link in the supplementary information these days. And you can go and look at how the data is fitted, makes it a transparent process in review, but also means that that data is cached in one place for goodness only knows how many molecules, with goodness only knows how many association constants, and linked within the literature. It's also, you can export this data as an Excel file, so it's in a readily accessible format because all this data is already curated in one place in the same format. The other thing is to develop novel antimicrobial technologies. As I'm often told, it would be handy to have more minimum inhibitory concentration results from multiple different molecules. The issue is that quite a lot of these data sets are held by drug companies or, again, splattered around the literature. But in more recent times, there's been a new initiative that's opened up at Public Health England, and this is an open innovation approach to discovering very early stage novel antimicrobial agents. And this is where I turned when I needed someone to give me a hand with figuring out if my compounds were antimicrobial agents or not. I didn't know anything about biology at this point, really, in the grand scheme of things. And these guys, they take your compounds and they will, they have a panel of antimicrobial resistant bacteria, and they will go and calculate your minimum inhibitory concentrations for you and work with you whilst not retaining any of your IP, which is lovely of them. They are fantastic human beings. Because they just want to see novel technologies come to the forefront, and they understand it's going to be individuals in small labs and small companies are probably going to do this, and they need a little bit of help, and handling antimicrobial resistant bacteria is not on everyone's training list. So they now have, I think it is a library of between 12 and 1500 molecules and their activities against a certain set panel of microbes. They've also gone and done some genetic figured out where there's resistance, the genetic code of these bacteria that are resistant, so they've got that data as well. And again, it's all held in one place, but not really taken advantage of as far as I knew last Tuesday. So again, could we use that data to add to the data that has already been produced by persons such as myself, to look at creating that big expansive data set that machine learning and artificial techniques need in order to help progress technologies. So it's with that I'd like to do a final thank you to the wonderful people within my research team. This is literally what happened on the day that we discovered that we had we can make gels in the lab. So this is one of my students, Jess. She was in charge of weighing out milligrams of compound endlessly into vials. Nia, who was responsible for dissolving these things in various aqueous salt solutions. Becca, who was responsible for timing a certain time to make sure that each vial had enough time to potentially gel. And then these guys here who were involved in making sure that gels had actually formed and then recording said data. So it really was a team effort, and it's a very expensive process in that way to get these little numbers in tables. And I'd also like to thank two of my chief collaborators and mentors and friends, Dr. Dan Mulvihill at School of Biosciences and Dr. Ewan Clark, who will never give me a decent photo of him at all, here at the School of Physical Sciences, and all my collaborators, Jose for helping me with the NMR work, and these body of funders without whom we wouldn't be able to do any research whatsoever. And it's at this point that I believe that is the last slide in my slideshow.